بسم الله الرحمن الرحيم. In our video today, we are going to continue our talk about innate lymphoid cells.
This is another diagram about the groups of innate lymphoid cells taken from another paper. It divided innate lymphoid cells into three groups. Group 1, which includes natural killer cells and innate lymphoid cells type 1, which depends on TBIT as a transcription factor and is stimulated by interleukin 15 and interleukin 12. They produce interferon gamma, granzyme, perforins, and they are responsible for type 1 immunity for intracellular pathogens. Group 2, innate lymphoid cells, includes innate lymphoid cells type 2, which depends on GATA3 as a transcription factor and is stimulated by innate interleukin 33, interleukin 25, and thymic stromal lymphoprotein. These cells produce interleukin 4, 5, 9, and 13, and they are responsible for type 2 immunity for extracellular parasites. Group 3 innate lymphoid cells includes lymphoid tissue inducer and innate lymphoid cells type 3, and they need retinoic acid orphan receptor gamma T as a transcription factor. They are stimulated by interleukin 1 beta and interleukin 23, and they produce interleukin 17, interleukin 22, tumor necrosis factor, and they are responsible for type 3 immunity for extracellular bacteria and fungi. And as we know from the previous video, lymphoid tissue inducer also is responsible for lymphoid tissue formation like Peyer's patches of the intestine. So again, for group 1, which includes natural killer cells and innate lymphoid cells type 1, they both need the transcription factor TBIT. They express it too. They promote type 1 immunity, which is critical for controlling intracellular microbial infections and stopping tumor development. Functionally, natural killer cells have a strong cytotoxic potential. They are responsible more for producing granzymes and perforins, whereas innate lymphoid cells type 1 produce a much higher concentration of interferon gamma than natural killer cells. And as I said in the first video about innate lymphoid cells, natural killer cells and innate lymphoid cells type 1 are considered the innate counterparts of CD8 positive cytotoxic T cells and T helper 1 cells respectively. So natural killer cells represents the innate counterpart of CD8 positive cytotoxic T cells, and innate lymphoid cell type 1 is the innate counterpart of T helper 1 cells. Innate lymphoid cells type 2 is dependent, as I said, on the transcription factor GATA3 and retinoic orphan receptor alpha, and they produce type 2 cytokines like interleukin 4, 5, 9, and 13 in response to tissue-derived cytokines interleukin 33 and 25 and TSLP, thymic stromal lymphoprotein. Innate lymphoid cells type 2 play protective role against intestinal parasites and have an important role also in tissue repair and could contribute to the homeostatic maintenance of the metabolism of adipose tissue in mice. From the bad side or from the other side, innate lymphoid cells type 2 can promote allergic inflammation at multiple barrier organs. Also, they can promote asthma-like lung inflammation in various experimental models. As regards the third group, innate lymphoid cells type 3 and lymphoid tissue inducer cells, they commonly depend on retinoic acid orphan receptor gamma T but their distribution and functions are different. So lymphoid tissue inducer cells mediate the development of lymphoid tissues during embryogenesis via the production of lymphotoxin. Innate lymphoid cells type 3 produce interleukin 22 that has been reported to directly act on intestinal epithelial cells to protect and mediate resistance to intestinal infections. Also, innate lymphoid cells type 3 play an important role in the regulation of the symbiotic relationship with the intestinal microbiota. Lymphoid tissue inducer were previously included here in the group 3 innate lymphoid cells because of their capacity to produce interleukin 17 and interleukin 22 as innate lymphoid cells type 3. Innate lymphoid cell subsets show a certain degree of plasticity, and this is the cause behind their heterogeneity, similar to T helper cell subsets. So innate lymphoid cells subsets can change their phenotype and functional capacities as a result of polarizing signals in the tissue in which the conversion occurs. Here is an interesting paper published in 2022 in Frontiers Immunology. Its title is Innate Lymphoid Cells More Than Just Immune Cells. In this paper, they said that innate lymphoid cells have unique features that make them stand out from the immune landscape. Innate lymphoid cells are relatively rare in lymphoid tissues and in the circulations, but reside mainly in non-lymphoid organs and particularly in the mucosal surfaces. So this is the first unique features for innate lymphoid cells. They also are recognized as a part of the innate immunity. However, they are not devoid of memory responses. Another feature, while other innate immune cells depend on pattern recognition receptors to initiate their activation, the role of pattern recognition receptors as toll-like receptors in the innate lymphoid cells is still unclear. Innate lymphoid cells have many receptors that can sense host-derived signals, for example, dietary metabolites, microbial products, hormones, neuropeptides, and cytokines, and so they can maintain the homeostasis of organs where they reside. So as regard the relation between innate lymphoid cells and the metabolism, they found that malnutrition and overeating, both of them, either malnutrition or overeating, will result in dysregulated innate lymphoid cells mediated responses. This is because of availability of specific dietary nutrients in one side, and also on the other side by disturbing homeostasis of adipose tissue energy stores, which expand or contract with nutrient availability. We can take vitamin A as an example of the effect of nutrients on innate lymphoid cells. Vitamin A, its availability will increase expression of gut homing receptors on innate lymphoid cells type 3 and type 1. On the other hand, it will make suppression on innate lymphoid cells type 2. Innate lymphoid cells type 2 in adipose tissue is very important for a process called beiging. Beiging is these are cells of multilocular adipose tissue cells from the white adipose tissue, which are capable of thermogenesis. So when vitamin A decrease or suppress innate lymphoid cell type 2, will decrease also the process of beiging. Again, vitamin A metabolite, retinoic acid, will promote the expression of gut homing receptors on innate lymphoid cells type 1 and type 3 and enhance innate lymphoid cells type 3 function. On the other side, retinoic acid suppresses innate lymphoid cells type 2, so innate lymphoid cells type 2 numbers will increase during period of vitamin A deficiency. So when we have increase in number of innate lymphoid cells type 2, we'll have increased efficiency of parasitic infection resistance. Also, as I said, innate lymphoid cells type 2 is important for maintenance of metabolic homeostasis by supporting type 2 immune environment characteristic of the adipose tissue of lean individuals. And dysregulation as a result of excess vitamin A as a nutrient, we will get obesity with persistent low-grade type 1 inflammation. Actually, innate lymphoid cells type 2 reside in visceral adipose tissue, where they are maintained by the expression of interleukin 33, and they produce interleukin 5 that support eosinophils, and interleukin 13 to polarize macrophages to the M2 phenotype and regulate adiposity and insulin resistance. As regards the role of innate lymphoid cells in tissue repair, we have innate lymphoid cells type 3 that produce innate interleukin 22. They make protection from tissue damage by their antimicrobial peptides and preventing any extracellular parasite or infection from attacking and making tissue damage. We have also innate lymphoid cells type 1 and natural killer cells. By their cytotoxicity and interferon gamma, they will make elimination of microbes and dying cells and debris. And the most important is innate lymphoid cells type 2, which produce interleukin 13 and 5, and they are responsible for tissue remodeling and repair. Another important role of innate lymphoid cells is through their communication with the nervous system. Actually, the regulation of immune responses has been shown to tightly connected to the brain and nervous system. And this happens through the autonomic nervous system with its neurotransmitters and neuropeptides, and also by the hypothalamic pituitary adrenal axis, which work in parallel to modulate inflammation and maintain homeostasis. In this diagram, we can see the different types of innate lymphoid cells with receptors to many neurotransmitters. Like innate lymphoid cells type 1 natural killer cells have receptor to glucocorticoids and epinephrine and norepinephrine. Also, innate lymphoid cells have receptors to vasoactive intestinal peptide and calcitonin response, calcitonin receptor like. And also in innate lymphoid cells, we have receptor to something called RET, and we have also acetylcholine receptors. It's also interesting to know this expression, neuroimmune cell units. Neuroimmune cell units means small cellular networks in which immune and neuronal cells are found at the same anatomic site. They interact with each other functionally and participate in tissue homeostasis and integrity. So this is the neuroimmune cell units. It's the unit which is composed from immune and neuronal cells that found are at the same location or same anatomic site, and they are communicating with each other to keep tissue homeostasis.
I hope you have enjoyed the video, and again I'm reminding you to subscribe to the channel and activating the alarming bell if you are interested in getting any new video, and thank you.